Well, hello again. This is lecture number 15, protein modification within the ER. As you can see on the diagram to the right, once a protein gets to the ER, there are a lot of modifications. Actually, that may not be very easy to see, but if you look at the membrane right here, this is the membrane. This would be the cytosolic side of the membrane. This would be the ER luminal side of the membrane. There are modifications that take place, and that's what we're going to be talking about in this lecture. This is a relatively long lecture, and the reason for that is I've really put all of the modifications together in a single lecture. And I'm not going to talk about the specific modifications on this slide because we'll get to those in just a second. Now, before we talk about these, I want to kind of tell you what the rationale is. We talked in the last couple of lectures about getting proteins to the ER, both to the ER lumen and to the ER membrane. Now, when you look at this diagram right here, you could say, okay, well, we've got proteins to the ER. There we go. So the next step is to look at this movement to these other compartments. Let's say the Golgi, the plasma membrane, lysosomes are outside the cell. Now that's all vesicular transport. We've made that point. And so it would be logical to talk about vesicular transport next. And in the past, that's usually what I've done. But every time I do it that way, I kind of feel like it's kind of out of sequence. And I'll tell you why. The reason is because once proteins get to the ER, a lot of things happen to them. And so in the past, what we've done is we've gone straight from protein targeting to the ER. And once we finished that, we talked about vesicular trafficking. But then we had to go back and pick up on these things that happen in the ER. So what I want to do this time is I want to say, okay, we've got the proteins to the endoplasmic reticulum. Now, before they leave that compartment, let's talk about what happens to them. Then we'll go back and we'll pick up on vesicular trafficking a little bit later on. So in other words, all those proteins that are targeted to the lumen of the ER, or those proteins that are in the ER membrane, those proteins that are in the ER membrane, the portion of the protein that's on the luminal side are going to be modified. So that's why we're really going to focus here on modifications to proteins in the ER. Then, as I said, we'll go back and pick up how those proteins move once they're modified. You'll remember this slide from lecture number 14 where we talked about conservation of membrane faces. So let me just put it to you this way. If this is the ER over here, now let's just say for the sake of discussion here, this is the ER. We'll just mark out Golgi here. Some of these proteins, as we saw, are going to be in the lumen. So there's a luminal protein. And some of these proteins, as we saw yesterday, are going to be transmembrane proteins. They could be top 1, 2, or 3, or they could be top 4, 4A or 4B. So here's the point again. Any protein that's in the lumen of the ER can be modified, and this portion of these transmembrane proteins can also be modified, that portion that's in the lumen. So that's what we're focusing on. What happens to these proteins while they're in the lumen of the ER? There are really three things that happen, and that's what we're going to talk about for the rest of this lecture. The first thing that happens is these proteins are going to get modified with carbohydrates. Carbohydrates are going to be added to these proteins, and they're going to be processed. Now, that whole process of adding carbohydrates to proteins, by the way, we're talking about sugars here. That whole process is called glycosylation. So we're going to glycosylate these proteins once they're in the ER. The second thing that's going to happen, we'll call that the first thing. The second thing that's going to happen is we're going to form disulfide bonds. And we'll talk about that. And then the third thing that's going to happen is that these proteins have to fold when they're in the ER. Now, remember, protein translocation into the ER is co-translational. The proteins, in other words, are not folded. So at some point, they have to completely fold so that they can achieve their native structure. So we're going to kind of lump folding and assembly of multi-subunit proteins together because that's kind of the same thing. It's our final three-dimensional structure, maybe a single protein or it may be a quaternary structure. So those are the three things that we'll be talking about. So let's start out by making a general point here. This doesn't have anything to do with folding, but when it comes to adding the glycolipids, or I'm not the glycolipids, but when it comes to adding the carbohydrates, in other words, the process of glycosylation and the process of disulfide bonds, all that's going to happen in the lumen of the ER. Now let's go back and look at this slide right here, this diagram to the right. We said before, if this is the cytosol over here, we said this could be the lumen of the ER. Okay, if that's the lumen of the ER, what we're saying is that the addition of the carbohydrates, which is shown here, oligo just means a chain of saccharides, refers to the fact that there's sugars, and the formation of these disulfide bonds are happening in the lumen of the ER. Now let's think about this in a different way, however. Let me erase that. And let's assume that this picture doesn't show the cytosol in the lumen of the ER, but rather it shows the cytosol and, let's say, the extracellular space. In other words, outside the cell. Now ask yourself, does that fit? And the answer is it does fit, because we know that when a protein, let's say a transmembrane protein, is inserted into the ER membrane, we know that the portion that's in the lumen of the ER, if that protein is incorporated into a vesicle and makes its way to the plasma membrane, we know that once that vesicle fuses with the plasma membrane, the portion that was in the lumen is going to be exposed on the outside of the cell. And so all of the modifications that we're talking about, the formation of disulfide bonds and the addition of sugars, are always going to be on the exoplasmic surface of the plasma membrane. Now you could say they're going to be on the exoplasmic surface of the ER membrane as well, right? Because think about the conservation of membrane faces again. Each leaflet of the membrane can either be the cytosolic or the exoplasmic. So the modifications that we're talking about in the ER occur on the exoplasmic side of the membrane, which means that when those proteins get to the plasma membrane, they are still on the exoplasmic side, which means they're outside the cell. Now, one other thing before we leave this says glycolipids. We're not really going to talk about glycolipids, but the point here is that lipids can also have sugars added to them as well. Okay, so glycoproteins are proteins that have sugars added to them. That's where we get the term glycosylation. In other words, we glycosylate those proteins and they therefore become glycoproteins. We can also glycosylate lipids and they become glycolipids. Again, our focus here is on glycoproteins. Anyway, the take-home message from this slide is just to recognize that these modifications take place in the lumen on the exoplasmic side of the membrane, and because of conservation of membrane faces, they're always going to remain on the exoplasmic leaflet of the membrane. Okay, so let's begin now by talking specifically about the glycosylation, or the addition of carbohydrates. If you look at this slide up here, you can appreciate how extensive this can be. In this slide, we've got the nucleus, so we'll color the nucleus in here yellow. There's our nucleus. You can actually see right here the nuclear membrane, the double membrane. And in fact, you can see it fold back on itself right there. Kind of hard to see what's going on right here. It looks like it might fold back on itself. This, therefore, would be the cytosol. And if you take a look at this cell, it's been stained with some sort of stain that will bind to and therefore identify carbohydrates. Now this is the plasma membrane right here. And you can see that in this cell, the plasma membrane is just coated with carbohydrates. And as we saw on the last slide, all these carbohydrates are outside the cell because they're attached to proteins or lipids, but we're focusing on proteins that were in the lumen of the ER and therefore outside the cell. That's also what this slide is showing you right here. In other words, this is a cartoon of what you're seeing above. In other words, all this kind of black fuzzy stuff here that we know as carbohydrates is showing up down here as these blue carbohydrate chains. Nothing really new there. It's just making that same important point. And you can see some glycolipids here as well. We don't want to forget they exist, but we're not really going to look at the mechanism because we're focusing on proteins. Now, the next thing we want to ask ourselves is, why would you add carbohydrates to proteins in the ER? What's that all about? Well, it really does four things for the protein or for the cell by doing it to the protein. And here's what happens. It aids in the folding of the protein. Now, what that means is that we know that a protein is going to fold spontaneously. Would it fold spontaneously without the carbohydrates? Yes. But if you remember when we were talking about protein folding way back in the beginning of the semester, we said that proteins can be modified. You can add an acetyl group or something like that, and that's going to change the way that protein folds. Well, that's exactly what's going on here. In other words, for some proteins to get the three-dimensional or tertiary structure that they need to function properly, they need to be folded in a certain way. We already know that. And the addition of carbohydrates is going to aid in proper folding of that protein. So they're important for protein folding. Secondly, they're important for stability. A lot of proteins are going to have a longer half-life. They're going to hang around longer. They're going to be more stable if they have carbohydrates added to them. So it's a way to lengthen the half-life of a protein. Thirdly, and this will be easy to appreciate given that last slide, they can be involved in cell adhesion. So think about that last slide with all those carbohydrates on the outside of that slide, on the outside of the cell. That's going to make the surface of that cell kind of sticky. And those carbohydrates can therefore play an important role in allowing cells to kind of hang together within a tissue. Now, we're not really going to talk about that. That's really outside the scope of the course. We're just focusing here on why we need to add these carbohydrates. And the fourth one is one that we can all relate to. Everybody probably knows what their blood type is, and that blood type is due to antigens on the surface of your red blood cells, and those antigens include carbohydrates that are added to proteins on the surface of your red blood cells. So in that case, what's going to happen is they're going to induce an immune response. In other words, if you get a blood transfusion and you get blood of the wrong type, then you can mount an immune response. I'm sure you guys have encountered that before and you know how that works. Again, we're not focusing on the immune response. That's immunology. We're just simply saying that if we turn it around, it's one way that the body can recognize itself by recognizing the appropriate carbohydrate structures on the surface of cells. So the bottom line here is that carbohydrates are going to do a lot of different things. Now, they're going to do other things in the cell as well. They're going to serve as a source of energy, for example, but we're talking specifically about why they're added to proteins. Okay, so let's kind of look at what these carbohydrates look like. They're going to fall into two groups. Let's start with the ones on the bottom. They're called O-linked sugars. They are small and simple. They're called O-linked because they're added to the hydroxyl group on a serine or a threonine. That's where they get the name O-linked. And we'll see this again on the next slide. The more common carbohydrates, and the ones that we will spend the most time talking about, are larger carbohydrates, much more complex. they're branched. They're linked to the amide nitrogen on asparagine. So here's an asparagine amino acid. They're going to be linked to that nitrogen atom right there. That's why they're called N-linked. So N-linked and O-linked refers to the atom on the amino acid side chain to which they're added. We can see that on this slide. We'll start again with our O-linked sugars. There's a threonine, so there's that terminal started out as a hydroxyl group, and we're going to add a sugar to it. In this case, the specific sugar that we're adding is N-acetyl-galactosamine. Now we'll see a lot of different kinds of sugars, N-acetyl-galactosamine, N-acetyl-glucosamine. We'll see different kinds of sugars. We're not going to, again, get bogged down too much in the kind of sugar residues. The thing you need to remember is these are going to be strings of sugars in most cases. So we'll take a sugar and another sugar and another sugar and another sugar. And so in this case, the first sugar that's added is N-acetylglucosamine, and then we have the remainder here of this oligosaccharide chain. Over here, here's an N-linked. There's that nitrogen atom right there on that asparagine side chain. The first sugar that's added in this case is an N-acetylglucosamine. Very similar sugars, but not exactly the same. Okay, and then you have the rest of the chain down here. So I like this slide because it really helps you to see, I think, very nicely what these sugars are doing in terms of being attached to the protein. We've got a covalent bond that's linking the sugar. A covalent bond that's linking the sugar to an atom on a side chain. Asparagine if it's N-linked, serine or threonine if it's O-linked. Okay, so now we're going to focus our attention exclusively on these N-linked sugars. So if we start out up over here, we can see our protein, and we can see we've got an asparagine right here. Now we don't see the side chain, but we know that side chain exists. Okay. There's a core region to this sugar tree. Remember, these are complex and they're branched. So as we'll see in the next couple of slides, there's a core region that consists of an N-acetylglucosamine, an N-acetylglucosamine, and then 3 mannose sugars. So if we just put a dot on each one of those, we see the same thing over here. So that's the core. There's the core. Now on the bottom, this is called a high mannose oligosaccharide because you can see to that core, we've added 3 more. Let's use yellow for these mannoses. We've added 3 more mannoses here. So that's called a high mannose oligosaccharide. On the one on the top, this more complex oligosaccharide, you'll notice that we've got a couple of more N-acetylglucosamines. And then we've got some other sugars here. We've got 3 galactoses right here. And we've got 3 N-acetylneuramic acids. 2 things that you need to take away from this. One is that every sugar, no matter what it looks like in its final stage, has this 5 sugar core. And we'll talk about that more in just a minute. Beyond that 5 sugar core, the actual oligosaccharide that's on the protein can be very different, as you can see right here. So high mannose oligosaccharides are going to have the core plus some other mannoses. Complex oligosaccharides are going to have the core plus maybe several other different types of sugars, and they're going to tend to be branched. Now, what you can see right here, shaded in gray, is the core. Now they've changed the color scheme on you, but you've still got 2 N-acetylglucosamines and 3 mannoses. So in gray, you've got your core. You can also see here that we've got some other sugars that are added. Now, what we saw on the previous slide was the final sugar. Now this gets a little bit complicated. So here's that was the final sugar, just so you could see what those sugars look like. Think about how the sugar is going to be added to the protein. You've really got two options. All of these are going to be these long oligosaccharide chains. This is sometimes also called a sugar tree because it's branched and it looks like a tree because it's branched like that. So you could add one sugar at a time. So in other words, imagine that you've got a protein in the ER and you add a sugar, then you add a sugar to that sugar and a sugar to that sugar, so forth and so on until you've got your tree. But you can imagine that that might take a lot of time. And we're going to see on another slide where that's not going to work for a couple of reasons. We'll get back to this issue. So what the cell does is it actually builds a precursor. It's a preformed precursor. So when we think about how these sugars are added to the protein in terms of the sequence of events, we've got to look at this preformed precursor. So what does the preformed precursor look like? Well, this is the way it's designated right here, because you have 3 glucoses. So let's actually do this a little bit differently. We've got 3. Let's start over here. No, okay, I'm sorry. We've got 3 glucoses. There are glucoses. Okay. We've got 9 mannoses. There they are. 1, 2, 3, 4, 5, 6, 7, 8, 9 mannoses. And we have 2 of these N-acetylglucosamines right here and right here. Okay. Now that's the way that sugar is designated. Now notice that with 3 glucoses, 9 mannoses, and 2 N-acetylglucosamines, you have your core. You've still got your core right here of your 2 N-acetylglucosamines and your 3 mannoses. It's just that in addition to that, you have 3 more glucoses, 9 mannoses, and 2, and you have the 3 glucoses, and you have actually 6 more mannoses for a total of 9 mannoses. You also have what's called a variable and non-variable region. It turns out that the core is called the core because it never changes. So the core is the non-variable region. The non-variable is the core. All these other sugars can change. And in fact, if you go back and look at the previous slide, you'll see that. Remember, both of those sugars, the branched complex one and the other one had the core, but then the other sugars were different. So those other sugars are the variable region and the core is the non-variable region. I also like this slide because, again, you can see really nicely the asparagine side chain, and you can see the sugar linked to that asparagine side chain. Now, let's think about how this is going to work. So it turns out that preformed precursor is made in the lumen of the ER, and we're going to talk in a minute about how that preformed precursor is made. But let's assume right now that it's made. And here's the ER, so we're over on this side. We're over here on this side. And that preformed precursor is anchored to the membrane. Now again, they've changed the color on you, and that's unfortunate, but there's N-acetylglucosamine, N-acetylglucosamine. There are your 3 core mannoses. There are your 6 additional mannoses, 1, 2, 3, 4, 5, 6. And then there are your 3 glucoses. So that's the core that we just described. It looks a little bit different, but the composition is exactly the same. And notice that it's anchored to the membrane by these 2 phosphates, which are anchored to this molecule called dolicol. Now, dolicol, as it turns out, is a byproduct of the cholesterol biosynthetic pathway. So as the cell is making cholesterol, there are some kind of offshoots of that, and dolicol is an offshoot of that. Here's what you need to know about dolicol that's important for our purposes right now, and that is that dolicol is a lipid. Okay, as such, it's nonpolar, which means that it's soluble in the membrane, because the membrane is made of these nonpolar fatty acids. Well, they're made of phospholipids, but the middle portion of that membrane is composed of these fatty acid chains. I know you guys have had that in other classes. We'll talk about that more, but we know that the membrane is nonpolar, the interior of the membrane. So this dolicol is actually links this preformed precursor to the membrane. You can see the same thing over here. That's what's being shown right here. Okay? Now, let's take protein targeting to the ER and link it with this. So here comes a protein. Now this protein, by way of review, what type of protein is this, by the way? It's not going to the lumen because it has an internal signal sequence. Is it a type 1, a type 2, a type 3, or a type 4? Well, as far as we can tell, it's not a type 4. We don't really know what's going to come up here, but let's assume that nothing else comes. So it's not a type 4. Okay, that's a type 2 because the amino terminus is out in the cytosol. Again, just by way of review, we're going to have our positive charges right here, right? Okay, so there comes that protein. It's moving through the translocon. The energy of translation is pushing the rest of this protein through the translocon. Linked to the translocon is an enzyme called oligosaccharyl transferase. It's going to do exactly what its name implies that it's going to do. It's going to transfer an oligosaccharide to the protein. Well, here's the oligosaccharide. The preformed precursor is sitting there right at the membrane. You can see why it's important that the oligosaccharide precursor is linked to the membrane, because that will allow it to be added to that asparagine in this case as soon as that asparagine emerges through the translocon. In other words, here comes the protein. There's an asparagine right there. Oligosaccharyl transferase recognizes that asparagine. It takes the preformed precursor, takes it off of dolicol, and adds it to the protein. And this is what's being shown over here. Here's your oligosaccharyl transferase. And notice that it has a binding site for the oligosaccharide. It also has a binding site for the asparagine. And so that's going to bring the two things together. The oligosaccharide is going to be transferred to that asparagine, and we've got a glycosylated protein. Now, before we move on, there are a couple things that we need to note. One, not every asparagine is going to have an oligosaccharide added to it. It has to be asparagine X and then serine, or asparagine X and then threonine. X can be anything except proline. So that's the consensus sequence. Remember, that word consensus means agreement. So that means every time you see an oligosaccharide added to an asparagine, there's agreement that this is what the sequence is going to be. Now, there's something else to note here. Not every asparagine that is part of this sequence is going to have a carbohydrate added to it either. The only way you know that is you look at a protein, and if you're interested in a particular protein and you want to know whether or not it has carbohydrates added to it, you can experimentally test it. And so you'll say you can take a look at the amino acid sequence of a protein, and you can say it has, you'll see this in the literature all the time, it has 10 potential N-linked glycosylation sites. That would mean that you have 10 asparagines that are part of a consensus sequence like this. You'll always see it written, though, 10 potential N-linked glycosylation sites. It means they all are in the right, are part of the right consensus sequence, but they don't always get glycosylated. I'm sure there's probably something else there has to be something else going on that determines whether or not it gets glycosylated. It's not a random event, because on a given protein, the same asparagines are always going to get glycosylated. But as far as I know, there's no way to look at the protein and predict it. You have to just go to the laboratory and experimentally test it. Okay? So, anyway, back to the main point here. The main point here is that asparagine gets glycosylated as that protein emerges into the lumen of the ER. In other words, as it moves through the translocon. Now that's important because we said that these oligosaccharides are going to play an important role in folding. Well, this protein hasn't folded yet because the whole thing hasn't even been made. So when this protein gets to the point of folding, the oligosaccharides are already there. So that's the role of oligosaccharyl transferase, a really important enzyme anchored to the translocon. It does the job of transferring the preformed precursor from the dolichol rather to the protein. Now, let's think about this preformed precursor a little bit. We know what it is, we know how it gets added, but we want to think about how it's synthesized, because how it's synthesized brings up some really important questions, fundamental cell biology questions. Technically, this preformed precursor is called a dolichol pyrophosphoryl, pyrophosphate, 2 phosphates, dolichol pyrophosphoryl oligosaccharide precursor. That's the big fancy name for the whole thing, the dolichol plus the 2 phosphates plus the preformed precursor. Now let's look at how it's made. Recall that the smooth ER and the rough ER are all linked together. Also recall that lipid synthesis occurs in the smooth ER. Also recall that this dolichol molecule, a byproduct of cholesterol metabolism, is a nonpolar molecule that's going to be in the membrane. So it can move laterally within the plane of the membrane. So that dolichol molecule is going to move laterally within the plane of the membrane and end up in the rough ER where protein synthesis is taking place. Now look what happens. You got the dolichol in the membrane. On the cytosolic side of the membrane, what happens is you add the first phosphate. Let's use purple for this. So we're going to add the first phosphate. Okay. Now this is cytodilyl triphosphate. Okay, so this molecule right here has 3 phosphates on it. One of those is going to be removed and transferred to dolichol, so now you have dolichol phosphate. And you're going to be left with cytodilyl diphosphate, because this one has 2. Okay? Now the next thing that happens is you take 2 uridine diphosphates. Okay, so this is a typical nucleotide, uridine. Okay, but it's a uridine diphosphate to which the sugar N-acetylglucosamine is linked. Now remember, N-acetylglucosamine is the first sugar on this preformed precursor. Remember, you have 2 N-acetylglucosamines, then 3 mannoses. That's your core. Okay, so what we're going to do here is take 2 UDP N-acetylglucosamines, and we're going to take one of those and we're going to take a phosphate off of it and an N-acetylglucosamine and add it to the phosphate that existed here. So we're going to take another phosphate and another N-acetylglucosamine. If we do that, we're going to end up with a uridine monophosphate. Now all we need is another N-acetylglucosamine. So from the second UDP N-acetylglucosamine, we don't take any phosphates. We just take an N-acetylglucosamine. That gives us our second N-acetylglucosamine, and what we have left over is a uridine diphosphate because we didn't need its phosphate. Now look what you've got. You've got dolichol pyrophosphate with the 2 N-acetylglucosamines. So we've got the beginning of that preformed precursor. Now don't lose sight of the fact that this is taking place on the luminal side of the ER membrane. And you might want to scratch your head at this point and say, well, that doesn't make any sense because this whole thing is going to be added in the lumen of the ER, and yet it's being synthesized on the cytosolic side of the membrane. Now, remember, the preformed precursor has 2 N-acetylglucosamines. It has 3 mannoses that make up the core, but it has 6 more mannoses for a grand total of 9 mannoses. So what's going to happen is that after you've added your 2 phosphates, your 2 N-acetylglucosamines, then 5 mannoses get added. And those are going to be added. We're going to have 5 guanidine diphosphate, another nucleotide here that has mannose attached to it. We're going to take the mannose off of all 5 of those and add them to the N-acetylglucosamine. So we've got 5 of the 9 that we need. Let me erase some of this. Now here's why this is so intriguing, and really, you know, it's a point to pause and think about how the cell does things. Because the question you might ask yourself at this point, as I said just a minute ago, is you scratch your head and say, why is this taking place on the cytosolic side? Well, once you get to this point, what you find out is that that whole complex that's been added so far, a large complex with 7 sugars, 2 pyrophosphates, flips in the membrane. So this is exactly what you've got over here, except now it's facing the lumen of the membrane. Now there are a couple of questions that you might want to ask yourself here. How does that flipping occur? And the answer is, we don't know. In fact, whenever you see something like this flipping in the membrane, and flipping is in quotations, that's a pretty sure indicator that we don't know how it happens. Now you might say, well, why would you call it flipping? Well, it starts out on one side of the membrane and then it's on the other side of the membrane. So somehow or another it got over there. So flipping is an appropriate term. We just don't know what that flipping event looks like. So not only is there some mystery surrounding the flipping, but the other question is this: why do you add 5 mannoses and then flip it? Why don't you add all 9 mannoses and then flip it? Or why don't you add all 9 mannoses plus the glucoses and flip it? Or we could go in the other direction and say, well, why don't you flip it before you add any of the mannoses? And we don't know the answer to those questions either. The easy way to look at it is to say that the cell's going to do what's energetically practical for it to do. So it must be energetically efficient to begin the process of building this preformed precursor on the cytosolic side of the membrane. It looks as if what the cell does is it builds as much of it as it can and then it flips it. Now all of these sugars are polar, so you got to ask yourself, how does this flipping mechanism get those polar sugars across this nonpolar membrane? And we just said we don't know that. But it could be that adding even 1 more mannose would make that impossible. Maybe if you have 6 mannoses, you can't flip it across the membrane anymore. So again, I love this because it shows you once again how precise the cell is. It's going to do what's energetically feasible, what's energetically practical. Looks like what it's doing is it's making as much of it as it can on the cytosolic side, because that's probably the energetically practical way to do it. Then it flips it over and now it's got to finish it. And so once it gets flipped over on this side, what's going to happen is we're going to have another dolicol in the membrane right here. Okay, now notice this dolicol is made from dolicol phosphate and GDP mannose. So it's made kind of like this one is made, except there's no N-acetylglucosamine there. So what do we have? We have dolicol with a phosphate and a mannose. And so what's going to happen now is this reaction is going to take place 4 times, in which we're going to take one of these, take the mannose off, and put it here. And when you do that 4 times, You've already got 5 mannoses there. We're going to end up with our 9 mannoses. Now all we have to do is add 3 glucoses. Same sort of thing is going to take place. The glucose donor is also made from adolyl phosphate, UDP glucose. That reaction is going to take place 3 times, and our 3 glucoses are going to be added. And now we've got our preformed precursor. 3 glucoses, 9 mannoses, 2 N-acetylglucosamines. Now if you go back to the previous slide, we know exactly now how that's going to be added. We're going to take oligosaccharyl transferase is going to take that preformed precursor and add it to an appropriate asparagine residue. Now, before we leave this slide, I just want to alert you to the fact that you need to keep your eye on these 3 glucoses. These 3 glucoses play an interesting role in this whole process. So I won't spill the beans right now. I won't tell you what they do. I'll just tell you, just kind of think about those 3 glucoses. Ask yourself this question. What are those 3 glucoses there for? Because remember back on that slide where we saw a high mannose sugar and we saw the more complex sugar, there were no glucoses in either one of those cases. So these glucoses are going to play a special role. We'll see what they do. Okay, so we know how this preformed precursor is made, and we know eventually how this preformed precursor will be added to the protein. Again, keep an eye on these 3 glucoses. Now, on this slide, we're jumping ahead a little bit, but I feel like it's important to kind of pause for a minute and see what's going to happen next. All the events that we've described so far, the making of that oligosaccharide precursor, adding it to the protein, we know is taking place in the ER. But what we've also seen is that the carbohydrates on proteins are very different. Well, the reason they're different is because even though the events that we've described are taking place in the ER, once those proteins are incorporated into a vesicle and move over to the Golgi apparatus, the Golgi apparatus is really going to modify those carbohydrates. Now, we'll talk about the Golgi apparatus later on, but the point that I want to make here is simply this. We've really got 5 compartments to the Golgi apparatus. We have the Golgi stacks in the middle called the cis, the medial, and the trans cisterna. Then we have the cis Golgi network, and we have the trans Golgi network. Now, again, I don't want to get bogged down on that here. We'll talk about it later. The point, though, is this. The cis Golgi network is the compartment of the Golgi apparatus that's closest to the ER. The trans Golgi is furthest away from the ER. And as these proteins that have had those carbohydrates added get to the ER, they're going to move through the ER on their way either to the plasma membrane, the lysosomes, or to be secreted from the cell. As they're headed to those potential destinations, they move through each of these Golgi compartments, all 5 of these Golgi compartments. You can't skip one. And notice that in each compartment of the Golgi apparatus, there are modifications to the proteins. I'm sorry, modifications to the oligosaccharides on the proteins. So for example, right here, we can get some phosphorylation of specific oligosaccharides. We get to this cis-cisternal, we get removal of mannose. We get to the medial, we can get removal of mannose, but we can also get addition of more N-acetylglucosamines. We get over here to the trans Golgi, addition of galactose, addition of a really complex sugar called abbreviated NANA. You get over here to the trans Golgi network, you can sulfate tyrosines, and you can sulfate carbohydrates. Now, the one thing that can't change is you can't change that core. Remember, the core doesn't change, but all those other variable regions can change. And in fact, if we go over here just to kind of see, kind of put this whole thing together, let's start out over here on the left. We can see our protein right here with our preformed precursor. We know that's the preformed precursor because we have our 2 N-acetylglucosamines, we have our 9 mannoses, 1, 2, 3, 4, 5, 6, 7, 8, 9, and our 3 glucoses. So after that event takes place, we're going to clip off those glucoses. There are 2 glucosidases in the ER that are going to remove those glucoses, and we're going to remove one of the mannoses. So those modifications take place in the ER. So what we're left with is 2 N-acetylglucosamines and 8 mannoses. 1, 2, 3, 4, 5, 6, 7, 8. Then all of this takes place in the Golgi. That's what we just saw on the previous slide. And this is where we begin to remove some more mannoses. We begin to add some different sugars. Here we're going to add a sugar. We can remove some more mannoses. We can add more sugars. You can actually tell how far in the Golgi apparatus this protein has moved by looking at its resistance or sensitivity to an enzyme called Endo-H. So Endo-H is an enzyme that will clip off some sugars. In fact, it'll clip off all the sugars. If the protein has moved to this point in the Golgi apparatus, if you treat that cell with this drug called Endo-H, it will remove the sugars. But once you get one step beyond that and you've added this next N-acetylglucosamine right here, it becomes Endo-H resistant. Now that may seem like a really picky thing, but it's a really important experimental tool in cell biology, because as people are looking at the movement of proteins through the secretory pathway, they often want to know, well, how far has it gone? And so you can't always say exactly, but you can say, well, it's Endo-H resistant or it's Endo-H sensitive. So it's going to tell you, roughly speaking, how far it's gone in the pathway. And then you can see what's going to happen is you've got your final complex oligosaccharide over here. Now again, this is all taking place in the Golgi, I realize that. But we've got to kind of put all this together. Okay. Now, let's head back to the ER and return our focus to the ER. And before we leave this slide, it's a good opportunity to ask a really important question related to these 3 glucoses. Now they become even more mysterious because you can see that they're removed. And if you look at the rest of this pathway, you can see that glucoses are never added again. So what's the point of those glucoses? Well, here's the point of those glucoses. Well, let me put it this way. I'm going to tell you what the purpose of the glucoses is, but then I'm going to tell you that a little bit later on, they're going to come back again. So here's the initial purpose of the glucoses. That oligosaccharyl transferase that's transferring this preformed precursor to the protein needs to make sure that it's transferring a completed precursor. So the thought is that these 3 Golgi residues are the sign, so to speak, that that precursor is complete. Those are the last 3 sugars added. So that's another way you can put it. If the preformed precursor has those 3 glucoses, then that means it's complete. And so once it gets added to the protein, we just get rid of them because they've done their job. Their job, again, was to signal to oligosaccharyl transferase, hey, I'm a complete precursor ready to be added to a protein. Don't forget them because they're going to come up again a little bit later on. But that's their job. All right, so that's it for carbohydrates. Let's shift our focus now to disulfide bonds. Just like with carbohydrates or oligosaccharides that are added to proteins, the disulfide bonds are also going to be added in the lumen of the ER. Consequently, any protein that has a disulfide bond in it that's at the plasma membrane, that disulfide bond is going to be outside the cell. It's going to be on the exoplasmic portion of the membrane. Now there are a couple reasons for that we're going to get into, but notice on this slide, it's because this is a reducing environment. What you're going to see in just a minute is this reaction that forms a disulfide bond is called an oxidation reduction reaction. So a reducing environment is an environment that's going to really keep the disulfide bond from forming. So not only out in the cytosol you don't have the mechanism for forming disulfide bonds, even if you had the mechanism, they would come apart just because the conditions aren't favorable for forming disulfide bonds. Now, I think you probably know what disulfide bonds are, but let's not assume that. Let's dive into the chemistry a little bit and see what these disulfide bonds are. Remember, cysteine is one of our special amino acids. Glycine and proline are special for their own reasons, but remember, cysteine is special because it's one of 2 amino acids that has a sulfur in the side chain. There's our sulfur. It's the only amino acid, however, in which this thiol group, this SH group, also called a sulfhydryl for obvious reasons, in which that group is a terminal group. Now the fact that that's a terminal group means that that SH group can do something that no other amino acid can do, and that is to form disulfide bonds. So we want to see what that's going to look like. So disulfide bonds are formed as a result of oxidation reduction reactions. Now on the next slide, we'll look a little bit more at the chemistry of oxidation reduction reactions. I know you guys probably know this, but again, to make sure we're on the same page. At this point, suffice it to say that if you have a protein, let's say you've got this protein up here, polypeptide number 1, which has a cysteine with an exposed sulfhydryl group. You got another protein over here, polypeptide number 2, which also has a cysteine, which is going to have an exposed sulfhydryl group. So you could create a disulfide bond between those. And a disulfide bond is simply a covalent bond between the 2 between the 2 sulfur atoms. So there's your disulfide bond, obviously called di-2 sulfurs. That's your disulfide bond. So that's a way to covalently link 2 proteins together. Now you could also have 2 cysteines with their sulfhydryl groups on the same polypeptide, and you could form a disulfide bond there. In that case, what you're doing is you're linking 2 different parts of the same protein together. This is called an interchain disulfide bond because it's between 2 different proteins. I always think of interstate. The interstate, like I-20, is an interstate. Interstates are called interstates because they go between states. It's I-20 whether you're in Louisiana or Mississippi or Alabama. An intrachain disulfide bond is within the protein in this case. Okay, now prior to forming the disulfide bond, these sulfhydryl groups are in their reduced form, which means they're electron rich, which also means they've got a proton attached. So it's going to exist as an SH group. That's the reduced form. Okay. In the oxidized form, notice that that hydrogen atom is gone. And in the reduced form, the region in the oxidized form, that hydrogen atom is gone because what's happened is we've created this disulfide bond in which electrons are being shared between this sulfur atom and this sulfur atom. Now, if electrons are being shared between those two sulfur atoms, then that means the electrons that were part of each sulfur atom, some of those electrons are now somewhere else. Because we can't keep those electrons and also share electrons like this. Then you would have too many electrons. So what's going to happen is electrons are going to leave these sulfur atoms and go somewhere else. And that's going to force the remaining, or that's going to force the sulfur atoms then to share the remaining electrons. So oxidation then, you may recall in a general chemistry class, oxidation is often, it's the oil rig thing. Oxidation is loss of electrons, and reduction is gaining of electrons. Okay. So in other words, when we're oxidizing these self-hydryl groups, what are we doing? We're losing electrons, and when you lose electrons, which have a negative charge, you're going to lose your protons as well. Now, let's look at that here. Again, here's the reduced form. Notice that in the reduced form, there is no disulfide bond. So an oxidation event is going to take place. So oxidation would be on this side, and now we're going to form a disulfide bond. Reduction would be on this side. Now let's use this example to the right to kind of illustrate again what's going on with oxidation and reduction, and introduce another really important part of this whole thing. Now, this example on the right doesn't have anything to do with the formation of disulfide bonds. It's just to illustrate what's going on with oxidation and reduction. So imagine that we've got this molecule right here. It's in its reduced form. Now it's in its oxidized form down here. Now that oxidation event in this case, we're not talking about sulfhydryl groups now, but in this case what happens is you're going to lose electrons. You're always losing electrons. Those electrons have to go somewhere. So in this case, those electrons that are being lost from this molecule are being added to this molecule called NADP. Okay. Now that NADP molecule is really important here because that NADP molecule is called an oxidant. An oxidant is something that facilitates oxidation by accepting electrons. So in other words, when we lose electrons from this version of this molecule, they got to go somewhere, and it's the oxidant that accepts them, or we could say the oxidant is responsible for removing them. Now notice what's going to happen here. Whenever you lose an electron, you're going to lose a proton as well. So this NADP here is going to pick up an electron. Okay. And actually, whenever you pick up an electron, you're going to pick up a hydrogen atom. We said we're going to lose a hydrogen atom. That hydrogen atom is going to neutralize the charge that came with a negative. So that NADP now is going to become NADPH. Okay. Notice also that we've got an excess hydrogen. Now I said we're going to lose protons. What I really haven't said is that we're losing 2 protons. And then you can see that right here. In that case, there's that proton and there's that proton. And over here, we're going to lose those 2 hydrogens. Okay? Now, just hang with me because you might be thinking to yourself, this is more chemistry than I really want to know. But you got to know the chemistry in order for the cell biology to make sense. Now, if you wanted to reduce or rather, if you wanted to transfer the electrons from that NADPH, you would need another oxidant, right? So in this case, the electrons are going to be transferred over to this molecule. Okay, so this is the oxidized version of this molecule, and when it picks up electrons, what's going to happen is we're no longer going to have a double bond here. We're going to have a single bond here, and we're going to pick up 2 protons. Okay, so in this case, this molecule is actually the oxidized form of this molecule is now going to gain electrons. It's going to undergo reduction, and here's the reduced form of that molecule. So the bottom line here is when you talk about formation of disulfide bonds, it's an oxidation reduction reaction. And when you talk about oxidation and reduction reactions, all you're really saying is we're moving electrons around. And as we move those electrons around, protons are going to follow those electrons. So if we have a molecule that's electron rich, it's in its reduced form. Whenever we lose electrons, it becomes oxidized, and when it becomes oxidized, we're going to form double bonds. Now, these events, these oxidation and reduction events, are going to stabilize the tertiary and quaternary structure of proteins, because as we saw on the last slide, we're able to form these inter and intrachain disulfide bonds. Now what we want to do is take the chemistry here and apply it to the cell, see what happens in the cell. So here's what's going to happen. Let's imagine that before I do that, let me just say that this is from a different textbook, not a textbook that I've used so far, but I really like this figure because I think it really makes it easy to see what's going on. So let's identify all the components. Here's our translocon. Here's our ribosome. There's our protein emerging through the translocon into the lumen of the ER. Now notice we've got 2 cysteines here. We have two thiol groups. Erase that. Okay, that's in those are in the reduced form. Okay, they're in the reduced form because they're in the electron-rich form. They haven't really given up their electrons yet. Now remember, in order to form these disulfide bonds, we need to lose electrons. And remember, if we're going to lose electrons, in other words, if something is going to be oxidized, we have to have an oxidant. Well, the oxidant in this case is PDI. And anything that that can function as an oxidant is necessarily going to be in the oxidized form. In other words, it's already going to have a bond, and we're going to break that bond by adding more electrons. So what's going to happen is the electrons that are associated with this sulfur atom and this sulfur atom are going to be lost, and they're going to be transferred over to PDI. And that's going to allow the formation of the disulfide bond between the sulfur atoms that are on the protein. Now, in that case, what happens is the PDI is now in the reduced form, and your disulfides on the protein are in the oxidized form. So all we've done here is we've moved these electrons. We've moved these electrons from the sulfur atoms over to the PDI. Now, it turns out PDI has sulfhydryl groups as well. And so in the case of PDI, it can function as an effective oxidant because the PDI is in the oxidized form, which means that you have a disulfide bond right here. And we're just going to break that disulfide bond. And we're going to break that disulfide bond by transferring electrons from here over to here. Okay? Now, this protein PDI is going to do more than that. So let's look at its name. It's protein disulfide isomerase. It's responsible for facilitating this oxidation reduction reaction so that disulfide bonds can form. But now let's think about this for just a second, and I really need a little bit more space to draw this, so I may draw this on the board when we get to class, but we'll try it like this. Let's say a protein, Let's draw the ER membrane here. We'll put the translocon right here. Okay. Here's the protein that's emerging through the translocon. Okay. This is the amino terminus. And we'll say there is a cysteine and there is a cysteine. If those were the only two cysteines on that protein, you would have no problem because we could form a disulfide bond and all is well and good. But imagine that there's another cysteine rather here, and maybe even another cysteine right here. So let's say we've got 4 cysteines. And let's also say, for the sake of discussion, that when this protein is completely folded, you need to have a disulfide bond not between this one and this one, but maybe between 3 and 4. Okay, so let's call this one 1, this one 2, this one 3, and this one 4. Okay, in other words, the cell doesn't really know that cysteine number 3 and 4 is coming. So PDI is going to do its job. It's going to form a disulfide bond between 1 and 2. But as the protein starts to fold, maybe that's not the most energetically or thermodynamically favorable conformation to that protein to have. So what's going to happen is PDI is going to facilitate oxidation and reduction reactions that allow the proper pairing of cysteine residues to create the proper combination of disulfide bonds. Let me draw this out a little bit differently. Let's say you got a protein that looks like this. Here's the N-terminus, here's the C-terminus. So we've got a cysteine here, 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 and here. Okay. If we form a disulfide bond between 1 and 2 and 3 and 4, we haven't really linked that protein together in any sort of way. But slide is getting a little messy. Let me get rid of this. What if we do it this way? What if we form a disulfide bond between these two and between these two? In other words, instead of forming a disulfide bond between 1 and 2 and another one between 3 and 4, we form a disulfide bond between 1 and 4 and 2 and 3. Now look what happened. If you do it like this, you've now got an effective way to keep that protein folded. And so that's what's going to happen. That's why this is called protein disulfide isomerase. Because the protein disulfide, the isomerase portion of it is responsible for allowing rearrangements to occur over and over and over again of these disulfide bonds until the most favorable combination has occurred, until the most favorable three-dimensional structure of that protein exists. Now let's think about this a little bit more, because there's a potential problem here. If we take those electrons off of the thiol groups on a protein and we put them onto PDI, on the oxidized form of PDI, so that PDI becomes reduced, that's all well and good, except what that means is that PDI can no longer accept any electrons. In other words, it's full of electrons. It's in its fully reduced form. So if you had a need to form more disulfide bonds on a protein, you would eventually run out of PDI that could serve as an oxidant. So what the cell needs is a way to transfer those electrons from PDI to something else. And it does that by transferring them over to a protein called ERO1. Now, another way to think about this then is that ERO1 is an oxidant. Okay, and ERO1 is going to oxidize that reduced form of PDI. In other words, it's going to accept its electrons. Now, when it does that, that's great because that regenerates PDI. It puts PDI back in the oxidized form so that it could pick up more electrons. So this ERO1 is really important. Don't worry about the diagram on this slide. I think it's really complicated. I actually pulled this out of the primary literature to try to show what's going on. Just get the concept here. But now if you think about this for just a second, we've got the same problem again, right? Because what happens if ERO1 fills up with electrons? If ERO1 fills up with electrons, then it can accept no more electrons from PDI. If it can accept no more electrons from PDI and you accumulate the reduced version of PDI, then it can accept no more electrons from the thiol groups on the protein, and you can't form disulfide bonds. So, you know what's coming. You got to have a way to get rid of those electrons on ERO1. Now again, here's why cell biology is so cool. Think about where those electrons might go. Can you think of anything in the cell that might really want to accept electrons and in so doing regenerate the reduced form of ERO1? How about oxygen? Oxygen is an electron acceptor. In fact, we know that oxygen is the final electron acceptor in the electron transport chain. So what happens if oxygen picks up electrons? Well, we know that if oxygen picks up electrons, and it will pick up electrons, it has a high reduction potential. Okay, that just simply means that if it's something that has a high reduction potential, then it's willing to accept those electrons. So if it accepts those electrons, then what's normally going to come with that, did we say? We're going to pick up two hydrogens. So if we have an oxygen atom that picks up electrons and then picks up two hydrogens, what do we end up with? Water. Now, is that a problem for the cell? No. The cell is 70% water to begin with, so that's not a problem. Now, you might say, well, we can't keep doing this and can't keep doing this, but the amount of water we're forming here as a result of this reaction is minuscule compared to the total amount of water in the body. So it turns out that we're going to take electrons, we're going to move them from the sulfhydryl groups over to PDI, from PDI to ERO1, from ERO1 to oxygen, where we create water. So this is the mechanism. This is all taking place in the ER. This is the mechanism in the ER for forming these disulfide bonds.